Hopefully you were able to come up with what I was looking for in that study question. If not, you'll see it appear shortly as we progress in this tutorial. All right, well, let's consider now what are some of those molecular signals that guide the growth of the axon, that influence the decisions made by that growth cone as it explores the environment through which that elongating axon is traversing. Well, we consider basically two classes of molecules. There are molecules that are associated with membranes of cells of the developing embryo. And these molecules may also be embedded in the matrix that lies between the cells. We call that the extracellular matrix. So these are bound molecules, cell-associated molecules, or extracellular matrix-associated molecules. That's one category. Another category are diffusible molecules. So as that implies, these are not cell-associated, they're not bound to a membrane or to the matrix, but rather they are freely diffusing in the extracellular spaces of the developing nervous system or the developing embryo. So let's look at specifically what we have in mind here. So again, these cell-associated molecules are on the surface of cells, or they're embedded in the extracellular matrix, and they mediate molecular interactions between the developing growth cone and the parenchyma, or that is whatever the substratum is that that growth cone is exploring. Now, the interactions between the growth cone and the surfaces can trigger second messenger systems within the growth cones that transduce extracellular signals into patterns of directed growth. Some of these cell-associated molecules play a role in stabilizing the linkages between axonal membranes and the structures that surround them. One important element will be the glial cells that begin to form myelin or begin to lay down a substratum over which that axon will be laid down. So there needs to be not just direction of growth, but also stabilization of the axon that is elongated in the wake of that growth cone. Now, the diffusible molecules, as I mentioned, they are secreted by target structures and diffuse into the extracellular spaces. So then by definition, the diffusible molecules can act over a much greater distance within the central nervous system or outside the CNS in the embryo than can the cell-associated molecules that are anchored to a membrane or the extracellular matrix. Okay, what I'd like to do next is to consider some specific examples of our cell-associated molecules to give you a feel for what kinds of actions they may mediate. So here's a figure finger text that just illustrates some of the important cell-associated molecules that we know about, beginning with those that are associated with the extracellular matrix. So here is a representation of that matrix, and the idea is that there are proteins and other kinds of molecules that present sort of a fibrous matrix in the extracellular spaces. And molecules can interact then with receptors, which are often the very same molecule. We call this a homophilic interaction because it's the same molecule interacting with itself. In this case, what we see are interactions among the integrin receptors. Now, these integrins themselves do not have enzymatic activity in the cytoplasmic surface of the molecule, but rather they interact with other kinds of molecules that do have that enzymatic activity. For example, kinases and other kinds of signaling molecules that can activate other second messenger systems for further signal transduction downstream. Well, those are the integrins. There's a variety of individual members of that family. Next, I'd like to mention the cell adhesion molecules. These are very important for mediating several different specific aspects of pathway development. We'll talk about some as we move along here. There are certain classes of cell adhesion molecules that don't require the activity or the presence of calcium. And so that's what's shown here in panel B. So these cell adhesion molecules, or CAMs for short, that are depicted here would be the L1 CAM as well as the neuronal cell adhesion molecule called NCAM. So these do interact with similar kinds of kinases and other effector systems within the cell that can influence transduction cascades within the cell that's interacting with these molecules. Now notice again, there's homophilic interaction of one kind of cell adhesion molecule with the very same type of molecule on the surface of another cell. Now there are cell adhesion molecules that are calcium dependent. They require calcium for their activity. Here's an example of one. It's called a cadhedron. So here we have cadhedrons that require the binding of calcium ions in order to engage their activity. And these activities then are mediated via a variety of second messenger systems, some of which can have positive effects, some can have negative effects depending upon the system in question. This particular cadhedron interacts with the beta-catenin pathway, which can have an impact on the assembly of actin filaments in the cell that is receiving this signal. And then lastly, I would mention the ephrins. So the ephrins are a very important class of molecules. The ephrin proteins are presented on the surface of cells, and they interact with receptors that we call Ephs, or ephrin receptors. These are tyrosine kinase receptors, and they can activate a variety of different kinds of second messenger systems within cells. Here we see an ephrin receptor interacting with the Rho GAP system that likewise can have an impact on the assembly or disassembly of actin filaments. Now I'll give you one example of the importance of ephrins and ephrin receptors in the development of circuitry. Let's consider, for example, the development of a topographical map. This requires axons from one kind of structure in the developing nervous system to project into a target structure, and to do so in an orderly way. We saw many examples of this as we talked through Unit 3 and considered our sensory systems, and perhaps we took it for granted that it is possible to establish such topographic maps connecting one station in a neural pathway with another. But it's really quite an amazing accomplishment in neural development. And for a long time, neuroscientists suspected that there may be chemical molecules that somehow are establishing the basic framework for a map, and then in subsequent stages of development, perhaps neural activity can be used to refine those connections. And indeed, that seems to be the basic paradigm by which topographic maps are established. We now know that ephrins and ephrin receptors play an important role in establishing topography in such systems. And here's an example that demonstrated rather dramatically the nature of the recognition between one supply structure and its target in the brain. And here what I'd like to present is a bit of a scenario derived from studies of an amphibian system, the frog visual pathway, which connects the retina to the frog's version of the superior colliculus that we call the optic tectum. And what's known about the anatomy of this system is that the posterior or the temporal part of the retina from the frog eye tends to send its axons into the anterior region of the tectum, preferentially avoiding the posterior region. Meanwhile, the more anterior or nasal part of the frog's retina sends its axons past the anterior part of the tectum to arborize mainly in the posterior part. Well, one fairly dramatic demonstration that there must be some kind of recognition that allows temporal retina to affiliate with the anterior part of the tectum and nasal retina to affiliate with the posterior part came from experiments that involved rotating the eye of a frog. Now, unlike mammals and many amphibians, if you destroy the optic nerve, it will actually regrow into the brain. And what was done in this experiment is that the eye was surgically rotated and then reattached. And after some time, the axons that were cut in the optic nerve were able to regenerate and reinnervate the brain. And so the behavior of the frog was then observed, and what was very clear is that after rotating the eye of the frog, its behavior was precisely off by about 180 degrees, which was the extent of the rotation. So a visual motor target appears to the upper left, and the frog protrudes its tongue to the lower right. Not good for the frog, maybe good for the fly. It indicates the identity of the retinal ganglion cells continued to be recognized by the appropriate domain of the optic tectum, irrespective of the surgical rotation of the eye. So what this experiment illustrated is that the anatomical structures of the eye remapped to the same divisions of the optic tectum, irrespective of the rotation of the eye. So in this case, we imagine that the fly is out in the temporal part of the visual field, which ought to be seen with a normal eye by the nasal part of the retina. The problem is that the eye has been rotated. So now what should be the temporal part of the visual field is being seen by the temporal part of the retina following rotation. Well, that part of the retina is going to innervate and project into this anterior part of the tectum, which is going to be responsible for inducing this reflexive motor action down into the right, which is now exactly 180 degrees off of where the appropriate strike should have been to acquire the target. So this experiment shows us that this mapping seems to be of a chemical nature rather than a functional nature, fully tuned and fully accommodated to the behavior of the animal. So many years had passed, decades as a matter of fact, that established this topographic map were worked out. So we know that in this system, what mediates the topography of this connection between the retina and the tectum are efferents and their receptors. So rather than there being a precise lock and key mechanism on a point-by-point basis, what we see is the establishment of gradients. Gradients of the ligand and gradients of the receptors. And the matching of ligand concentration to available receptor is what establishes the continuity in the topographic mapping, such that the most posterior temporal portion of the retina is mapped to the most anterior portion of the tectum. And then a gradient is established for the rest of the posterior temporal retina. And that gradient then is what allows for the exclusion of axons that then come to be mapped into the posterior part of the tectum, those axons being derived from the nasal part of the retina. So efferent signal is critical, we think, for the establishment of topographic maps, not just in the optic tectum of the frog, but we think wherever we find topographic maps in the developing central nervous system, including ours.